I'm Kelly Gillespie. I work with the Missouri Biotechnology Association, your statewide life sciences organization. We're made up of universities and medical schools and large and small companies that use biology or biotechnology to produce a product or service. And we also have an open and welcoming door to all the ancillary and really necessary supportive companies that make up the people engaged in the life sciences, ranging from law firms to construction companies to venture capital firms. And we are so pleased that you're part of the MOBIO community and that we can bring you this MOBIO Breakthrough Series to allow our members to get together. And want to thank Washington University in St. Louis for their engagement, their leadership with MOBIO. And to that point, Robert Blaine from the School of Medicine is MOBIO's key point of contact at WashU to MOBIO and serves on our executive committee. Would you say hello, Robert?
Absolutely. Again, Kelly, thanks for the invitation. My name is Robert Blaine. I'm the Assistant Vice Chancellor for Medical Public Policy at Washington University School of Medicine. And what we're going to talk about today are the sort of things that make me excited to do my job every day, to be quite honest with you. And we're clearly moving into a challenging time with the COVID-19 pandemic, but what gives me encouragement is the tireless work happening not only by our physicians and other healthcare workers, but by our researchers who, frankly, even when the pandemic first really started to rear its ugly head last spring, stepped up and continued the work, shifted a lot of that work to focus on this new disease. And we're now starting to see the products of that work in, again, record-setting timeframes. So again, thank you, MOBIO, for hosting this conversation today, for Dr. Curiel for being part of it, and looking forward to getting into it. So thank you.
Robert, thank you for being with us this morning and facilitating in this entire program. You've provided really important feedback to us as we shifted into some more virtual offerings for MOBIO to keep engaged with our membership. One of the key elements of MOBIO is maintaining that statewide voice for science and business in our Missouri State Capitol with both the legislative branch and the executive branch. It is one of our absolute primary functions and part of our mission. We are thrilled to see several elected officials join us on today's call to learn and to hear from Dr. Curiel. And so I want to thank, I see Senator Jill Shoup with us from St. Louis County, longtime friend of MOBIO and someone that has been on our bio benchmarking program. I saw Representative Phil Cristofanelli. I believe Phil is from St. Charles or St. Peters, and really pleased that he was able to join us as well. I think Stephen Roberts also registered. I don't see him having signed in quite yet. Is there anyone else that I'm missing that if you'd speak up, I really do prefer to give recognition to folks. Well, our folks that offer themselves up for public service to serve on the board of directors for the state of Missouri are tasked oftentimes with a lot of attention in the press and public for the campaign side of service and the political, hyper-political nature of how they're treated in the media. But this is emblematic of the kind of the learning that has to go on, that they avail themselves up, that they would sign on to today's program and learn. Today, we're really pleased to offer up Precision Virologics for the spotlight and the focus. Dr. David Curiel is the current CEO of that startup that has been described in the press as an underdog competitor in fighting the vaccine, COVID-19 vaccine. But the opportunities that his technology and his co-founders are bringing to it could really not just help with COVID-19, but in many other ways. And so David also serves as the director of the Biologic Therapeutics Center at Washington University. And you guys can find him on LinkedIn or on WashU's website. So with no further ado, David, thank you for your time today and being with us and with our MOBIO membership.
Sure, I appreciate the opportunity. Everybody can hear me? Wanted to thank the organizers. Your outreach is amazing, and the local flair aspect, that's what hooked me. I'm celebrating my 10th anniversary as a St. Louisan. I live on the World's Fair grounds. My house is full of World's Fair paraphernalia. I'm a huge fan of the city. In addition, your outreach was so effective, the number of people. My wife is adjunct faculty in the Brown School, so I've watched her give these lectures frequently. And so I'm impressed with her numbers, and I know that I need to top the number of attendees she's gotten, and so far it's looking good. First slide.
I've noticed that she has signed on, David, and what she doesn't know is I've got a secondary MOBIO waiting room that is about triple full of attendees.
So I'm going to talk to you about Precision Virologics, a St. Louis startup, and I'm going to speak on precision adenovirus engineering for optimized vaccine outcomes. And I think you'll see the story has a nice local angle. Next slide. So as background, I don't need to educate you on how terrible the COVID-19 pandemic has been this year. There have been more than 50 million cases recorded worldwide. In the US, there have been more than 10 million cases. There's high morbidity, high mortality, and the economic and societal needly, as well as mental health aspects, have been catastrophic. Next slide. So logically, this has driven unprecedented efforts to develop effective vaccines. The US government has advanced support through established vehicles. The Department of Defense, BARDA, the NIH, and others. In addition, there's been specific high priority funding through Operation Warp Speed that we've all read about in the paper. As well, there's been unprecedented investment from big pharma companies, many with vaccine programs, many for whom COVID represented their entree into the field. Next slide. So recently, there's been a lot of press about some clinical success in phase 3 trials. I'm showing you here New York Times headlines from last week relating to Pfizer's phase 3 trial, but these same slides would be equally applicable to this week's headlines with Moderna. And that is to say, for both of these vaccines, phase 3 trials showed more than 90% effectiveness. As background, these are a new class of vaccines. They're mRNA vectors, a nucleic acid that's complexed with what's described as a lipid nanoparticle. Both trials completed phase 3, and these are the most advanced clinical trials to date. So is the vaccine race over? Next slide. Next slide, yeah. So this was the headlines in the New York Times the same day that the positive data was revealed. Don't get too excited about the coronavirus vaccine from Pfizer or Moderna. I'll tell you why. And one of their things in that same issue was 11 points to consider. And within that article, they even noted that these results actually gave hope to the other vaccine efforts, and they were not at all a call to desist. So let's look at why that should be the case and what additional niche opportunities might exist for newer vaccines. Next slide. So the Moderna and the Pfizer vaccines share mRNA as their vehicle, and they also must be delivered intramuscularly. There's something unique about the way muscle takes up nucleic acids that's not operative in other delivery schemas. So if there are other ways that you could deliver that would give you a better response, this vehicle won't let you do it. In addition, there's been a lot in the press about the instability of mRNA. This is a major logistical problem. These need to be stored at -70 or -20 degrees, Pfizer and Moderna respectively. And this challenges not only third world cold chain, but United States. Cold chain is the preservation of vaccine potency from the manufacturer to the patient. And it embodies the trucks, planes, freezers that are required to do that. In addition to the unprecedented demands of this species for cold chain, it's difficult to make mRNA. It's synthesized chemically, it's not upscalable, and it's challenging capacities. So despite the clinical success, there's some logistical and practical limits with this vaccine technology. Next slide. So if you're thinking about another vector, an alternative vector, what should that vector embody? What are the critical requirements that make this more facile, more implementable, more accessible? Well, the first is you'd want it to be deliverable to body sites or body contexts that will give you an optimized immune response. Intramuscular delivery isn't the best way to derive immunity in the GI tract or in the lung. So you'd want a delivery schema that gave you the optimized immunity where it was relevant. You'd also want a vaccine that was cold chain compatible. I cringe when I read about these special packets that Pfizer is going to need to ship this vector at -70 degrees in the United States. One can't even imagine how that's going to be accomplished in the third world, which after all could remain a repository of disease if we don't eradicate it there. In addition, you want the optimal potency, so you're not as required to make so much. It's a basic business concept. Next slide. So at Precision Virologics, we're advancing a technology that we believe can address those requirements. The vehicle is adenovirus. This is a viral vector. It's the agent of the common cold in humans that's been used for more than 50 years in military recruits when they're mustered into the service with an enviable safety record. In addition, it's a tool molecular biologists like. It's the most highly characterized virus in the scientific armamentarium. We know a lot about it. We can engineer it rationally. In that regard, we can modify the virus, and this engineering can change key functionalities relevant to vaccine utility. Next slide. So at Precision Virologics, we've worked hard to develop novel adenovirus vector technology to make a vaccine company. And we've recognized that vaccines are of high interest to big pharmacy now. Recently, GSK, Johnson & Johnson have made major adenovirus acquisitions. In this regard, we possess unique technology that we think will make us a logical partner for the many big pharma companies now interested in the adenovirus space. Next slide. So what are these technologies? What do we have that nobody else has? Well, we use simian adenovirus as a lead agent for COVID. So just like human adenovirus has been used as a vaccine, we can modify the adenovirus that's normally infecting chimpanzees. What's unique about this is sufficiently different from human adenovirus that it will traverse preformed immunity. So this will enable us to give the vaccine to all people, irrespective of pre-exposure to adenovirus. Mike Diamond and I evaluated this technology via the intranasal route, and we showed immunity that was distinct in sterilizing the upper respiratory tract, and this represents a lead agent, which I'll tell you a bit about. But going back to my requirements for the best vaccine, what adenovirus can do that mRNA cannot do is be delivered by the respiratory route to elicit optimized respiratory immunity. Next slide. You're going to feel like I've gone to the St. Louis Zoo in this part of the talk. The other agent that we've explored is the adenovirus that normally infects sheep, ovine adenovirus. And this is a virus that has been employed previously for gene therapy and vaccine applications in humans. It has a track record of regulatory approval in the US and Australia, and there are methods to upscale it for human use. Importantly, the ovine adenovirus embodies unique biological facets. It has a unique thermostability that makes it even more cold chain competent than regular adenovirus, and substantially more so than mRNA. And at Precision, we presently have an NIH-funded program exploring ovine for an infection called chikungunya. Next slide. And lastly, we have a technology to achieve optimized potency. And that is to say, the dendritic cells are key immunoregulatory cells in the body that are critical to orchestrating the optimized vaccine effect. We have adenoviruses, or we've been able to engineer adenoviruses with enhanced delivery to dendritic cells, and therefore optimized potency. This optimized potency reduces the production burden required for vaccine platforms. Next slide. So I just want to remind you, based upon the positive outcomes of Pfizer and Moderna's vaccines, we've now been able to contemplate their limits. And we're able to conceptualize what the best vaccine should look like. It should have delivery for optimized immunization. It should have cold chain compatibility. It should be optimally potent to minimize upscale challenges. Next slide. So our technology at Precision addresses these requirements. We have simian adenovirus, for which we have a pending patent. It's been licensed. And this addresses the issue of optimized delivery for optimized immune response. Importantly, soon after the publication by Mike Diamond and myself, Bharat Biotech, the largest producer of vaccines in the world, licensed this vaccine to implement human trials in Asia, and they're on track for a phase 1 trial in India by the end of this year. Precision has licensed rights from WashU for US, Europe, and Japan, and we're exploring partnerships. We have the patent for ovine adenovirus, and as well, we have an issued patent for DC-targeted adenovirus, which has been in-licensed to Precision. So I'm going to tell you that other companies have also made major investment in adenovirus, and within that space, Precision's technology platform is unique, and I'd assert addresses the critical requirements for an optimized vaccine. Next slide. So we're not the only people that have been interested in adenoviruses, but I'd say we've been interested in them longer than anybody. Within the COVID space, major pharmaceutical companies are advancing COVID-19 programs. CanSino in China, AstraZeneca, Johnson & Johnson, and the so-called Russia agent. Johnson & Johnson is predicting 9 billion doses of their virus. These are trials in phase 2 to 3, and adenoviruses have never been compared head-to-head with mRNA vaccines, but what we do know is they're unrivaled with their ability to induce antibodies and T-cell immunity. Next slide. So within this context, we have unique technology for adenovirus vaccines for forward-going work, but right now we have a lead agent ready to be advanced as a competitor to the RNA vaccines and to the other adenovirus programs. We have a simian adenovirus that traverses preformed immunity. With Mike Diamond, we've designed a unique stabilized SARS antigen. We uniquely use an intranasal administrative route, and we've achieved sterilizing immunity, clearing the upper respiratory tract, which has been a unique and advantageous facet of our vaccine compared to others. Next slide. So I don't like the word underdog. I'd say alternative vaccines. Our approach was considered significantly different but interesting, that Nature did an editorial that highlighted that the first vaccines out of the block may not be the best, and that some of the newer technologies, ours included, may end up being the best. And people always invoke Sabine and Salk. Salk invented the polio vaccine. We all take the Sabine vaccine. Next slide. So Precision Virologics is a local story. We benefit from the rich infrastructure in St. Louis. Cortex provides this growing and vibrant entrepreneurial culture of wet labs and cores. And now that they've got this great hotel, it's just amazing. It also provides critical networking and makes connections for expertise and funding. And we also have links to the rich academic infrastructure. Washington University's Drug Development Center provides expertise for advancement and development of lead agents. So again, we couldn't have done it without this local support, and hats off to Cortex and BioSTL. They do an amazing job. Next slide. In addition, we have the ability to leverage our investments by utilizing in-place NIH mechanisms. And there are NIH mechanisms that provide leverage support for GMP production, that foster advancement to the FDA. And one of the advantages of my training, I spent 5 years in the NIH, is I know my way around the place, and that has helped to leverage funding. Next slide. We've really benefited from advisors, and I'd highlight the local angle. Mike Diamond is a world expert in developing diseases and emerging infections, and his models have fostered rapid evaluation of our vaccine candidates. Dan Hoft runs the Vaccine Treatment and Evaluation Unit at SLU, one of only 5 such units in the country, and provides a base to endeavor novel phase 1 trials. I made a beeline to his office when I arrived in St. Louis 10 years ago, We have a number of co-funded grants together and trials together. He's been a rich resource. Dan Streblow at the Vaccine Institute in Oregon is an expert in chikungunya, and Jerry Both in Australia is our expert in ovine adenovirus. So we've covered the waterfront with critical expertise. Next slide. So I'm currently the interim CEO and founder of Precision. I've had two successful previous companies, Autoimmune, which is a vaccine company, and Dionatrix, which is a company that uses adenovirus for cancer therapy. And I'm showing you the second edition of my book on adenovirus. I wrote the book. I'm now doing the third edition. And Brad Keller has extensive experience in the Cortex district with drug development and leveraging forward new agents. And he is our VP for preclinical development. Next slide. I can't overstate the value of SLU's Vaccine Treatment and Evaluation Unit. This provides direct support for new phase 1 trials. It provides networked support to the NIH, and I've worked closely with their center in advancing our programs in chikungunya and now in COVID. And again, a real resource for the area. Next slide. So we don't think COVID is going to be the last pandemic by a long shot. And we think our technology has proved its mettle. It was the lead agent that eradicated Ebola in West Africa 4 years ago. And it was waiting in the wings for COVID. And within a month, you'll hear the results from J&J and from AstraZeneca that will highlight adenovirus's place within the COVID armamentarium. And I've told you about our technology and how it will build upon that. So our plan is to continue the development of our lead agent, exploit NIH and BARDA resources, and use this as a mechanism to adapt our technology to other emerging infections. Next slide. And I'll take your questions now. Thank you.
Dr. Curiel, it's we all that have been around know that collaboration is a team sport, and that's how things get done. It is really exciting to see that that major opportunity for the powerhouses at Washington University and St. Louis University, and their prestige in the Center for Vaccine Development and the senior scientists that they have accumulated there, to see those working together and as part of the St. Louis life sciences ecosystem, it brings a lot of pride to my heart. I know that people work really hard at collaborations, and it is not easy. When the rubber meets the road, the science has to prevail, and the science has to be top notch. And great to see that happening on your end. No question, just a comment.
Okay.
Any other questions from others?
Kelly, I'll just kick things off. Dr. Curiel, again, thank you for the presentation. I wonder if you could just expand a little bit more and define sterilizing immunity and what that would mean, comparing what a patient who has an intramuscular or the shot in the arm vaccine would experience when exposed to COVID versus somebody who potentially benefits from an intranasal.
Well, that's a great question. Well, first I just highlight the practical facet. Intranasal vaccines are easier to take, and single shot vaccines have a much higher compliance rate than vaccines that require multiple administrations. So the simple fact is the way we give that vaccine will engender a higher level of compliance than the current RNA vaccines. But in essence, when a vaccine's given, you develop the hallmarks of immunity, antibodies and cell-mediated immunity. and that would prevent a infection from propagating in your system if you were infected. That doesn't necessarily mean that virus resident in parts of your bodies has been eliminated. So you could be protected from severe disease, but you could still be transmitting the virus. Sterilizing immunity, by virtue of the fact that you induce the immunity in the upper respiratory tract, provides protection and eradication. So thereby accrues the possibility of eliminating lateral transmission.
Thank you.
It's interesting how, what did Caesar say? He said that all progress in society derives from war. Many of these themes existed in the vaccine field, but COVID brought them to fruition. I didn't want to fully disparage the mRNA approach, because 20 years ago, I was actually the first to show that it worked, and I liked it back then, but back then I saw the practical limits of it. So COVID has driven a way to address those limits. Intranasal vaccination isn't new. It's been existing at a low level, and I've had programs in it in influenza and anthrax, but COVID has driven it to the fore. So in some ways, many themes of progress have been driven by the demands of the COVID pandemic.
I had a question. And this is Mark Shisley at St. Louis University. I do not work in our vaccine development center, so I don't have any knowledge to share there. This is more of just a general question, and maybe I missed this. Did you mention the timing on this of when this might be available? And
no, it's
I'm sorry.
No, it's a great question. Go ahead.
And then just to follow up to that, after that is given the urgency in the United States to get a vaccine out to the public quickly, do you view this as a viable option in the U.S., or is this more of something we'd look to distribute in other parts of the world that may not have the abilities we do to have the cold, the storage ability?
It's hard to know. When we started later than the big programs, and so we knew there would be a logistical issue as to whether we would be implemented no matter how superior we were. So our goal was simply to show the science and see where it fit within the armamentarium. I mean, literally, MOBIO does stuff that's so real time. Literally this morning, I was looking at estimates from an Indian company to produce my virus that put us in a timeline of the first clinical trial by the spring of next year. So that's the soonest we can envision doing it. But your question's a good one. But what vaccinologists always hearken back to is the Salk and Sabine paradigm, where the first vaccine that came out and was seen as the miracle people were waiting for wasn't the one that had the practical benefits that made it the ultimate winner and the one that we all take. So I think within the vaccine world, there's a sense that the best agent will emerge, and probably in relatively short order. I just despair of the public and how they understand this, because I'm a virologist and a vaccinologist, and if you read the newspaper last week, you would have thought Pfizer had solved it and there were no problems. So this week, you would have thought it was Moderna had solved it, and by God, there were problems with that Pfizer approach, and they were being spelled out. So I told my wife, I said, next week when the adenovirus trials are released, then you'll get the full story on the problems with the RNA approach. So it's a incremental education, but I hope this has helped.
David, maybe I'll just comment too on Mark's question. We're really at what I would call preclinical development. We still have to go through all the GLP toxicology and then get into the clinic. So, we're a ways away, but, we would push as fast as we can, but we still, for IND submission, we have to go through the standard development processes for for a vaccine.
I have a question. This is John Ruman with BioRecruiting Solutions. Dr. Curiel, from a business perspective, how were you able to participate in the Warp Speed funding with this project? And does the venture capital community showing interest in these secondary approaches to vaccines with the coronavirus?
So by the time we entered the field, a lot of the Warp Speed money had been allocated, and that was just a fact. And in the past, we've used a kind of leveraged approach to move forward. NIH support and then getting pharmacy partners. And so far, that's what we've pursued. We've written a number of grants for COVID. We're waiting for their outcome. But no, we didn't benefit. We have right now 2 pending BARDA proposals, and one has been embellished as a dossier on Senator Blunt's desk. So we've tried to enter into the fray, but we started later than others. And the second front, we've enjoyed lots of interest from industry, and we're having discussions with logical partners that we hope will move this forward.
This is Jill Shoup. I have a several part question, if you don't mind. So first of all, is it possible that more than one vaccine could move forward and there would be options available based on different needs? Or is that just untenable in terms of the marketplace? That's my first part. My second is, we talk about inhaling or the respiratory inhaling, your option. What about young children? Do we envision them being provided a vaccine? And if so, how would that work in your case? And my final part of the question is, do you envision this to be a seasonal vaccine, or could there be a one and done kind of vaccine that would create an immunity for the rest of your life, as we used to see? Or do you envision this mutating and needing to be updated on a regular basis?
Those are all good questions. In the first regard, it's absolutely possible that there will be a range of vaccines that are context relevant. And let me give you an example. The intramuscular injection has an age dependency in terms of how efficacious it is. So there's not enough data yet with mRNA to know how that will play out, but it's not illogical that one route would be better in one age group than another route. And there's precedent for that. But we certainly know with this new vaccine class, nucleic acid vaccines, Moderna, Pfizer, there's some concern about how the elderly will respond based upon the issues of intramuscular delivery in older subjects. So I think there will be more than one vaccine, more than one route. The second question related to children. I think there's very little data on how these vaccines will perform in children, and how that data will be acquired will be really interesting. But all of these trials are based on very limited patient subsets, and there's no approved trial with a nucleic acid vaccine right now. So this is new turf. There's no approved trial with adenovirus vaccines yet. So there's not the large number of patients that would tell you how to address children. So open question. And the third one was really interesting. With each microorganism, the degree to which antibodies or cell-mediated immunity is critical is biologically dependent. And the positive facet about the T-cell immunity, the cell-mediated immunity, is that's the long-lived immunity. So that would give you the possibility of one and done. So the agents that require induction of antibodies for protection, like influenza, those are the ones that require annual vaccines. Some organisms where you get T-cell immunity, it's one and done. There's a growing body of evidence that T-cells may be very important in SARS-CoV-2. So it may be that as we move forward, it will evolve to a one and done. I will say that one of the advantages of adenovirus that I push in a more scientific talk is the ability of adenovirus to induce much better T-cell response than nucleic acid vaccines do.
Rob Rose is right here. I got a quick question for you about autoimmune response.
Okay.
RNA, I know that RNA is, it's not been implemented in this way before.
Kelly, I can't hear him.
Can you hear me?
Now, yes.
Sorry about that. Just really quick about autoimmune response. What do you, foresee, since RNA vaccine like this is brand new, never been used before, people suffering from diabetes, MS, other, underlying health issues, what do you think would be the impact on those folks that are dealing with those already established autoimmune issues?
No, that's a great question. So, about 25 years ago, people were using plasmid DNA vaccines, and there was tremendous interest in them, tremendous entrepreneurial investment, but their efficacy in humans proved to be less than desired, and they were abandoned. So they've now come back as mRNA vaccines. At the time of the DNA vaccines, these same issues of autoimmunity were discussed, but in the limited phase 1 trials that were done, that never presented as an issue. So that's given the companies like Moderna and Pfizer some level of comfort. It remains to be determined, but the precedent in the DNA vaccine trials suggests it isn't a showstopper.
Thank you.
Does anyone else have a question they'd like to ask? Dr. Curiel, do you have any final comments? We're right on our schedule and doing great, even with a robust set of Q&A. Anything else you are thinking you'd like to point out?
I don't want to sound like just an inveterate St. Louis promoter, but what I will say is that we have real richness here in emerging disease models, in vaccine evaluation, in viral vectors, and by any standard, the ambitions to create a defense vaccine site or defense center make all the sense in the world, because there's many facets here of tied together could make our region a leader in this area.
And some of these great dreams take a long time to come to fruition, but you've hit on that opportunity that we need to be thinking of as Missourians of where do we set ourselves apart? Where do we position ourselves in when there's a new federal offering? These are not the type of projects that are funded for 6 months and go away. When we've had a chance at competing for the National Renewable Energy Lab in the 1970s, well, you know what? It's still in Colorado 35 years later. And when we had a chance at the NREL or the NBATH biodisease lab that ended up in Manhattan, Kansas. We're glad it's in Manhattan, Kansas, but it sure would be great if one of these big ones lands in St. Louis, and it will land there because not for political reasons. It will land there because our science is so very strong and sets itself apart. That's the key part. I'm really pleased that this breakthrough was able to touch on such a timely topic. As I remind everyone, the MOBIO Breakthrough Series is recorded. It is on our archives. So if you have colleagues or peers that you believe should see this session and see Dr. Curiel's presentation, go to the mobio.org website and you will find it. And just like you could go back in August, we had Dr. Rick Nettles from Johnson & Johnson, talking about their COVID approach. As Dr. Curiel mentioned, did you say 9 million or 9 billion doses?
9 billion.
Yeah. Once we get into the illions, I stop hearing too well. But 9 billion doses also kind of with a lot of bases on the adenovirus approach. And so MOBIO heard that from their J&J senior medical director back in August. And we love how these stories keep coming together and the intranasal aspect. I've had several member companies that are now very interested in what they can do on nasal washes and mouth washes as prevention opportunities, and that they're looking for consultants to get them into the game and into the funding mix for this. And so you see an entire industry shifting to not just be profitable, but to be helpful in the United States and the world being better than its current condition. And I want to just thank everybody. I did notice that Representative Stephen Roberts has joined the call from St. Louis. I announced that earlier, but Stephen, thank you for joining.
Glad to be here. Sorry for being late. Thank you so much for putting this together.
You bet, sir. You bet. Okay. Stay posted. We will announce the next MOBIO Breakthrough series soon. We will have this coming month BSA Life Structures be our MOBIO Member of the Month on our website. Avail yourselves of that opportunity as well, and keep coming at us. This is a member-driven organization and a member-driven programming. So thank you for those people that nominated Precision Virologics to get some really well-deserved attention today. So everyone have a great day, and we'll see you soon.